Okay, so guys, grab something you can write with. Grab something you can write with, and let's take a look at this lab. So, as we just discussed, looking at homework, guys, we're talking percent yields. And as we talk about percent yields, the difference between what we did in homework and what you're about to do is that you're going to be generating your own numbers. You will not be given a starting point for your theoretical yield calculation. You're going to figure out the mass of one of the reactants that you started with. The other one will be excess. Then, guys, you will not be given your actual yield. You will actually grab a hold of one of your products and you will weigh that, and that will be your actual yield. So guys, everything that you learn to do, thank you, Dylan, everything that you learn to do in homework, you're going to actually be doing in lab. So with that said, guys, take a look down here at the bottom of the lab. So guys, looking at this balanced equation, Bailey, be good. So guys, looking at this balanced equation, this is the reaction that you're actually going to be carrying out in lab. And I think you would agree that to be successful, you need to understand what this reaction is all about, and then specifically which portions of it are we going to focus in on as we do our calculations. So guys, with that said, let's start sort of high order in terms of understanding this. Because you know about 5 different reaction types: single and double displacement, decomposition and synthesis, and combustion. Which one's this? Guys, it's actually none of them. This is not any of the types of reactions that you've been shown, which shouldn't be surprising to you because, guys, you understand the 5 that you know are the tip of a really big iceberg. There are hundreds and thousands of classifications of reactions, and you only know 5. So, guys, this is not any of the reactions that we know. It doesn't mean we can't study it. It just means you don't know the type. If you're curious, this is actually what is called a weak base, strong acid neutralization reaction. And we're actually going to study this after spring break today, or spring break next week. So, but guys, with that said, we can still pick it apart. So guys, let's take a look at the reactant side of this reaction first. So one of these might be vaguely familiar to you. The other one is probably unfamiliar to you. So guys, let's talk. HCl. Do any of you make the connection? What is HCl? Hydrochloric acid. So guys, let's talk. You've seen it in lab, the last lab we did. Guys, what does HCl look like? It looks like a liquid, but it is not a liquid because it's something dissolved in water, which gives it what subscript? Let's write it down. Now, guys, sodium bicarbonate, I would be surprised if you know what it is until I tell you what it is. Do you know? It's baking soda. Yeah, so guys, sodium bicarbonate is actually baking soda, and it is very basic. And so baking soda is a white powdery solid, and its chemical formula is sodium bicarbonate. There is actually baking soda in baking powder, but there's leaveners in there as well. So guys, we've got baking soda, which is a solid. We've got hydrochloric acid, which is aqueous. Now guys, when you mix these together, you'll notice that we're going to be generating 3 products. So let's talk about what they are. And what's the subscript for water? It's a liquid. Now guys, carbon dioxide, I think you're familiar with. What is the subscript for carbon dioxide? It's a gas. So guys, based upon the fact that one of your products is a gas, what will you experience as you add the acid to the base? Bubbles. As this goes on, the carbon dioxide will bubble away as a gas. So now guys, let's talk about our last product. So our last product is salt. Solid, liquid, gas, or aqueous? Here's the trick. Guys, clearly salt is not a liquid and salt is not a gas. But what does salt do in the presence of water? It dissolves. And notice, guys, there's a lot of water around. There's the water that the HCl is dissolved in, and there's the water that the reaction is producing. And this table salt will dissolve into all of the water that is present in the reaction. So guys, now let's talk specifically about what you're going to do in lab. And we'll look at the procedure in a minute, but guys, here's how this is going to go. You are going to start with approximately 1 gram of sodium bicarbonate. That is the reactant that you will have a fixed amount of, making it which reactant? The limiting reactant. Now guys, here's the trick, and you're going to be held accountable for this. You don't have to have exactly 1 gram, but you should not have less than 1 gram. It's got to be about 1 gram, but more than 1 gram. So as you're weighing out your sodium bicarbonate, I would encourage you to make note to yourself somewhere on the lab, if you don't think you'll remember, it's got to be about, but not less than 1 gram. Does that make sense? Okay, so now guys, as you add the acid to the sodium bicarbonate, you are going to be turning your reactants into basically carbonated salt water. Literally, when you're done with this reaction, you're going to have a dish full of carbonated salt water. If everything was clean and you did it perfectly, you could drink it. You've just made carbonated salt water. But guys, here's what we're going to do. We are going to do a percent yield for the product that is easiest to isolate. And guys, the product that's easiest to isolate is the salt. So this is what we're going to do the percent yield for. That's our X. So guys, if that is the thing we're doing the percent yield for, how do we get rid of the carbon dioxide? It does it for us. It just bubbles away. Guys, how do we get rid of the water? Boil it off. And where have you learned that we boil salt and water apart to keep the salt? What piece of lab equipment do we use? An evaporating dish. So guys, we are going to do the entire lab in an evaporating dish so that we can simply then boil away the water and collect the salt in the dish. But guys, when you boil salt water, as it gets dry, what does the salt start to do? Fly out of the top, right? And before, guys, we were just like, no big deal, we'll make a mess. Why do we care about salt flying out now? That's your yield. Every time you see a little chunk of salt go, woohoo, your yield just went down. So what are we going to do about it? We're going to put a lid on it, which we call a watch glass. And guys, remember when we use a watch glass is a curved piece of glass, and we put it on there concave up rather than concave down, so the water drips back in rather than running out the sides. So guys, do you understand what we're up to? Okay, so let's look at the procedure. Read over this with me. There are a couple things you want to write down. So step number 1, clean with water and dry an evaporating dish in a watch glass. Just get it real dry. Then number 2, weigh them both. Then number 3, add approximately 1.0 grams of sodium bicarbonate to the dish. Now guys, you're going to do this, right? More than 1 gram. Not a lot more, but it's got to be more than 1 gram. By the way, guys, why do we care? Why can't we be slightly less than 1 gram? What does that do to your calculations? You lose a significant digit. See, guys, if you have less than 1 gram, you don't have a digit in that ones place, which takes 3 significant digits and turns it into 2. So we're going to get more than 1 gram, giving us a 3rd significant digit. Okay, so reading along then, guys, step number 4, re-weigh the dish and the watch glass with the sodium bicarbonate. Now guys, step 5, we need to talk about this carefully. Once you've done that, it says react while stirring. You may want to underline that. Guys, make sure that you're stirring the reaction as you go. So react while stirring the base, the sodium bicarbonate, with excess 1 molar HCl. Guys, what does it mean that the HCl is excess? You got all you want. You got a big old squeeze bottle. Guys, you're going to have all the acid that you need. But now read the last two words of step 5. Until complete. Guys, how do you know when the reaction is over? It stops bubbling. Here's what you're going to see. You're going to have a little pile of sodium bicarbonate in the dish, and you're going to start adding the acid drip by drip, and these drips of acid are going to land in the pile of sodium bicarbonate, and it's going to start to foam. And then eventually you're going to add so much acid that there's now so much water present that the sodium bicarbonate will dissolve into the water, and it's going to disappear. But it's still there. It's just dissolved. And then, guys, as you add more acid, you're going to see you're going to go drip, and it's going to go fizz, drip, fizz, drip, fizz. How do you know when you're done? You're going to go drip, no fizz. That's when you stop. Don't go on. Drip, no fizz, you're done. You know that you're now out of sodium bicarbonate, and the reaction's over. So then, guys, reading along, step 6, isolate the salt by evaporating the water with the watch glass on there. Capture the salt. Then 7, after it's cool, reweigh everything. And then, guys, rinse up, clean up. And, guys, please do not put the watch glasses in your drawers because I need them for 3rd period. You guys understand? Questions about the procedure? You okay? Okay, so guys, I'd like to get you started on the calculations. Hey, John. What is that? Oh, no, thank you. I know. I've let you down. Thanks, though. I think I just crushed Mr. Coxon. Okay, guys, let's talk calculations. Guys, calculation number 1 says this. It says, calculate the mass of sodium bicarbonate that you began with in the reaction. How much should it be? About 1 gram, not less. But it's not going to be 1 gram. But we need to know exactly what it is. So how do we find the mass? Subtraction. So guys, we know, but I'll show you to remind you, we know the accepted way to communicate mass by difference. How do we do it? We identify the things that we're subtracting. So guys, we have the mass. Feel free to write this down if you want. We have the mass, and guys, I don't know what's wrong with my board. The tracking down here is a mess. There's nothing I can do about it. Because we have the mass of the dish and the watch glass and the NaHCO3. But we want the mass of the NaHCO3. So what do we have to subtract away? The dish and the glass. Minus mass of the dish and the watch glass gives us the mass of the NaHCO3. Can you picture it? Just like we've been doing since August, right? And then, guys, we're going to do the subtraction and go on from there. Then on the back, you will be calculating your actual yield, your theoretical yield, and your percent yield. Then, guys, 3 conclusion questions. Number 1, what are possible reasons why you didn't get 100% yield, above or below? If yours is low, explain it. If yours is above, explain it. Number 2, what's up with this idea of limiting and excess reactants? And then, guys, number 3, in theory, why is it impossible to have more than 100% yield? But what explains it if you do get more than 100% yield? And guys, we talked about that in class last time. Yeah?
So, when we're evaporating out the water,
No, and that's the point. So if you can picture it, you've got the dish, right? And you've got this, and you're going drip, fizz, drip, fizz. When you go drip and there's no fizz, that one last drop of hydrochloric acid that you added converted that last little bit of sodium bicarbonate into salt water. And so when you've done that, let me zoom out. Oh, it's actually right here. When you've done that, this is all gone, right? You have added exactly enough of this to make this all go away. So actually, when you do that drip, no fizz, there's none of this. There's also none of that because you've added exactly enough to convert this into these things. So the only stuff that is in your dish, assuming that your dish was clean, is salt and water, and the carbon dioxide's gone. So there is no HCl in there. That's why you don't want to just go and squeeze a bunch of acid in there, because you're going to add more than you need, and then there would be acid in there. But if you add that one drip that doesn't fizz, you've done it perfectly, and there isn't any acid in there. It's all been converted. So, you guys good? We got it? Okay, goggles first, guys. There's bottles of acids at your stations. Oh, you guys are awesome. Thank you. We'll see you next door. I knew I could count on you because I would have forgot.